Mary, our next speaker is Liz Bryda from University of Missouri, a cross-species novel genetic cell ablation technology involving CD59 and Intermedilysin.
That title seems so long. So I want to, well, you should have waited for it to come up. So I want to thank the organizers for inviting me to speak today. And I'm going to tell you about our new targeted cell ablation system that we've been developing across species for several years now. Targeted cell ablation is a very valuable approach to study in vivo cell functions during things like organogenesis or tissue homeostasis or regeneration. And we use it to understand cell lineages and developmental processes and disease mechanisms. So some of the features that you want to look for in a good cell ablation system include that it needs to be specific, it should be efficient, you don't want it to have off-target effects, it should be effective in whatever cell type you want to study, and ideally it should be inducible. And as many of you know, we have lots of methods to do this currently, and they include things like microdissection or laser-based techniques. We have certainly a lot of genetic approaches where we make transgenic animals that you can then either directly or indirectly target your cell of interest for ablation. And we use chemicals and antibodies as well. And I've listed some of the very common cell ablation methods that are out there now, including things like diphtheria toxin, HSV, thymidine kinase, and nitroreductase. But as many of you are probably aware, all of these have some issues, whether it be a problem with bystander or off-target effects, or that they don't work rapidly. So we were looking for a system that might get around some of those issues. So our cell ablation system, we call it H for human, CD59 ILY. CD59 is a widely expressed protein that's it's a cell surface glycoprotein, and Intermedilysin, or ILY for short, is a bacterial pore-forming toxin. And ILY binds human CD59 very specifically. And as you can see in this cartoon, once ILY binds to CD59 on the surface of the cell, it oligomerizes and inserts into the membrane and ends up poking a hole in it, forming a pore, and the cell will lyse. So to try to test use this interaction to come up with a new cell ablation model, my colleague Shubin Shin, who at the time was at Harvard, made this transgenic mouse model. And what he did was he wanted to put the transgene expresses the human CD59 on the surface of erythrocytes. And then he administered ILY, and it led to lysis of red blood cells. And so this was published, it was a beautiful paper published in 2008 in Nature Medicine, and I can't show you all the data, but I want to summarize some of the things he found by using that particular model. We learned that ILY binds very specifically to the human version of CD59. And if you look over here, he looked at a number of different species, because a lot of species will express CD59, but ILY is specific for the human version. This is a system that can work on cells that aren't synthesizing protein, which is an advantage over some of the other existing systems. It had no effect on wild-type mice, so ILY is not toxic to the mice, even at very, very high doses. It's safe to work with because humans naturally have antibody to this bacterial toxin. He was able to demonstrate that lysis is dose-dependent and it's very rapid, and that you can do repeated dosing, and that it's very versatile in terms of how you deliver the ILY. So he showed it could be done by IP, IV, and IM. So we wanted to go on and make an inducible model. And we also wanted to demonstrate that you could ablate cells in a solid organ. So the mouse model that was made next is the one shown here, where we floxed a stop in front of HCD59. And that way you could mate this particular animal to the Cree animal of your choice, where you had Cree driven by whatever promoter you want. So that in these animals that result from that cross, you would get HCD59 expression exclusively in your cell type of interest in the presence of Cree, and then you could give the ILY injection, and that should lead to cell lysis in your cell type of interest. So I'm going to show you some of the data that was just published in JCI just recently. And this is for liver. So in this case, we were interested in ablating epithelial-based cells in the liver, and there's two types, hepatocytes and biliary epithelial cells, or BECs. And so in this first set of experiments here, we simply took those inducible HCD59 mice and mated them to a Cree driver that was where Cree was being driven by the albumin promoter, and that will express Cree specifically in hepatocytes and the BECs. And so we could show that, sorry, this mouse is sticky. We could show over here that we did get HCD59 specifically in the cell types of interest, and then upon administration of ILY, you could track the ablation of those cells by following either serum ALT levels, which is indicative of hepatocyte damage, or you could follow it looking at bilirubin, which was indicative of ablation of the BECs. The next set of experiments, we wanted to, again, look at specificity. So here, that albumin-driven Cree was delivered by a viral vector directly into to target the hepatocytes. And again, we could show that expression was specifically in the cell type of interest, and again, by measuring the specific type of damage that was being seen once you administered ILY, it was specific for what you would expect for ablation of hepatocytes. And down here in this final experiment, we mated the inducible mice to an inducible tamoxifen-driven SOX9 Cree line. And here they gave the animals tamoxifen that turned on the Cree. You got HCD59 expression specifically in the BECs. And when you administered ILY, you could show that there was damage just by this increase in bilirubin. So to sort of summarize what this all showed is that, yes, we could ablate cells in a solid organ. It was very specific. So we wanted, of course, because I work in rats now, we wanted to show that we could do this in rats. So we basically took the same construct that had been used in the original mouse model and did the same thing in a rat. And again, we're able to demonstrate that you could get very rapid lysis upon administration of ILY of the red blood cells that were expressing the human CD59. And this is now a nice model of anemia, and you can see that the animal is very anemic based on this. And this is, again, we did the same, wanted to make an inducible version of this in the rat. So ours is basically the same construct. It's a floxed stop HCD59, but we also added ZS green, so we had a reporter. And so in this case, we mated it to, there aren't a lot of Cree driver lines in the rat right now, so we had access to one where the Cree was being driven by the tyrosine hydroxylase promoter. So this will express Cree primarily in the TH positive neurons in the brain. And so we were able to demonstrate by mating these animals and looking at the double hemizygous animals that we were in fact getting co-localization of both ZS green and HCD59 in a portion of the brain where we know there's a high percentage of TH positive neurons. And then we went on to do ICV injections of ILY, so administering ILY directly into the brain. And we're in the process of doing a number of characterizations, but we did some behavior studies. And this, again, for those of you who are familiar with the Morris water maze, you're just basically putting the animal into this pool of water and wanting it to find a platform using distal tools. So it's sort of it's a behavioral test for memory and learning. And what we could show is at the end of the training session, when we removed the platform and asked the rats to basically remember where that platform was, that the animals that were expressing HCD59 on those TH positive neurons and that had been given ILY clearly had some deficits in remembering where and getting to the platform. Okay. So we wanted to now, so that's great. So we've shown that we can use this in rodents, but we wanted to extend it to other species. So in collaboration with Suman Gurung and Anand Chandrasekhar at University of Missouri, we've been trying to look at this in zebrafish. And as you might imagine, because the system developed in a mammalian system, we've had to do a lot of optimization to sort of make sure that we don't destroy the activity of the ILY, but have conditions that are conducive to working with zebrafish. So again, we think we've kind of got that under control. And I'm going to show you one of some preliminary data in one of our studies, where here we're just injecting the messenger RNA directly into the embryos. And it's either messenger RNA with ZS green and human CD59, or a version of that where we've mutated the bases that are within the ILY binding domain. So this should be a non-functional, not able to bind ILY. And we have, of course, controls, which are important. And so the experiment is basically to inject the embryos, look for the embryos that are expressing ZS green, then treat the embryos with ILY, and then start to monitor them for cell lysis. And so again, this is very preliminary data, but what we could see was even as early as 1 hour after treatment, we were beginning to see death in the embryos that should be expressing the human CD59 after they've been given ILY. And this is a picture from 4 hours post-ILY treatment, and you can clearly see that this embryo is not morphologically normal, and is probably there's probably cell lysis going on. And we followed a subset of these animals out to 24 hours post-ILY treatment. And in the group that were expressing the wild-type version of human CD59, we had 100% mortality. And we didn't see death in any of the other groups that we looked at. So we think this is pretty promising, and we're very excited about further optimizing this for use in zebrafish. So to summarize, this HCD59 ILY cell ablation system, we think it's specific, it's efficient, we haven't seen off-target effects. It appears to be effective in all the cell types that we've studied. We now have inducible versions, and it's very versatile, and we hope that it has the potential to be used across species. So with that, I'd like to thank my collaborators and acknowledge my funding sources, and I'm happy to take questions.
Questions?
I think your rat study might have answered my question, but does ILY cross the blood-brain barrier?
So that's a really good question. In the original work, they showed that it did, but what they were seeing it in was macrophages in the brain. So they think the resident macrophages were picking it up and going into the brain. When we did the mouse studies, they were able to show that you could administer it IP. They did do a neural model in that paper. And it wasn't as effective. So I think it is having trouble crossing the blood-brain barrier. Shubin is working on maybe doing a recombinant ILY that will be able to cross the blood-brain barrier. So the answer is yes, you have to do much higher doses. Thankfully, that doesn't kill the animals, but it's not quite there yet.
Very good.